investigate without the...
We have to read back to determine that it's
We went for half a meal and I kept just waiting.
Order in the court. Today, we're investigating the death of cell 3104. Prosecution, please present your case.
Your Honor, we've seen these cell death cases 1,000 times, and the culprit is always apoptosis.
Wait, Your Honor, we're missing something. Look, there's iron accumulation, reactive oxygen species, and lipid peroxidation.
There's only one type of cell death that could have done this.
Ferroptosis!
You may be wondering how Ferroptosis, our mystery killer, carried out its evil deed. Well, Ferroptosis is a form of iron-dependent cell death that occurs when the cell is unable to control the accumulation of lipid peroxides in the cell membrane. To start the story, we first need to introduce molecules called reactive oxygen species, or ROS for short. These are highly reactive molecules derived from oxygen, such as hydrogen peroxide, superoxide, and hydroxyl radicals. A major source of ROS in the cell is the mitochondria. This organelle is responsible for oxidative phosphorylation, which produces most of the cell's ATP, a source of energy. Inside, electrons are passed through a series of proteins along the inner mitochondrial membrane called the electron transport chain. Normally, in the final step, electrons are transferred to oxygen to form water. However, electrons can sometimes leak out early, which leaves oxygen incompletely reduced, forming superoxide and other ROS. Small amounts of ROS can be handled safely by the cell. However, when they start to build up, these molecules can cause cellular damage, which can lead to ferroptosis. So, what happens during ferroptosis? First, we need to look at the cell membrane. It contains a type of lipid called polyunsaturated fatty acids, or PUFAs for short. These lipids are particularly vulnerable to reactions with ROS as they contain some relatively weak carbon-hydrogen bonds. This means an ROS can remove a hydrogen from these lipids, forming a lipid-centered radical, which reacts with oxygen to form lipid peroxide. This initiates a chemical chain reaction called lipid peroxidation, a defining characteristic of ferroptosis. Lipid peroxides steal hydrogens from neighboring PUFAs, creating more and more lipid peroxides that eventually compromise the integrity of the membrane, leading to cell death. But there are more accomplices in ferroptosis' wicked plan, notably iron. Labile iron is highly reactive and can convert hydrogen peroxides to hydroxyl radicals in a process called the Fenton reaction. Similarly, it can convert lipid hydroperoxides back into oxygen-centered radicals. These molecules are highly reactive and can both initiate and help propagate the chain reaction. Thus, ferroptosis is said to be iron-dependent. So, how do cell membranes remain intact when ROS are present? Luckily, the cell possesses several defenses to keep lipid peroxidation in check. With the help of a molecule called glutathione, the GPX4 enzyme detoxifies lipid peroxides by converting them to non-toxic alcohols, stopping the chain reaction and thereby preventing ferroptosis. In addition, the body maintains iron homeostasis by regulating absorption and storing excess iron in non-toxic forms, such as bound to the iron storage protein ferritin. Ferroptosis is implicated in a wide range of diseases. In Alzheimer's disease, iron accumulation in the brain promotes lipid peroxidation, triggering ferroptosis. Similar findings have been observed in other neurodegenerative diseases, including Parkinson's and Huntington's. In ischemia reperfusion injury, oxygen deprivation, followed by its rapid return, causes oxidative stress, leading to ferroptosis. An emerging application is as a cancer therapy. Inhibition of GPX4 leads to buildup of lipid peroxides, sensitizing cancer cells to ferroptosis. This has prompted research into the selective induction of ferroptosis in cancer cells. This novel therapeutic approach holds great potential for treating cancers, including drug-resistant and metastatic cancers. Overall, there is still much more to learn in the fascinating field of ferroptosis.
It seems to me that ferroptosis is the true killer. Case closed.